Hey everyone, Dr. Steve here. You know it as well as I do, but most students need a B or better in A&P. But did you know that students are doing that all over the US using my Anatomy and Physiology Digital Suite as a low-cost alternative to expensive textbooks? Digital Suite delivers the key concepts of A&P with short, assignable tutor videos and a bulleted narrative study guide that not only explain the concepts of A&P, they show them happening. Combined with state-of-the-art dissection and physiology lab simulations and a vast question bank for quizzes, homework assignments, and exams, Digital Suite meets students where they are and gives them the tools to master the content. It's the perfect supplement to an OER textbook or just on its own. If you're an A&P instructor, contact your McGraw-Hill representative today and ask about Anatomy and Physiology Digital Suite. If you're a student, tell your instructor about it. There's a link to Digital Suite in the show notes of this episode. Get your B or better in A&P with A&P Digital Suite. Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, where I've been teaching A&P since 2002. I'm sitting in my office space in my house, which has now been rendered into a quarantine room for the oldest of my 3 cats. My cat suffers from hyperthyroidism, which means that his thyroid is producing way too much thyroid hormone and making his cells way too active. So he's burning a ton of calories and losing a lot of weight. He went from 15.5 pounds to 10.5 pounds in about 3 months. That's a huge amount of weight loss, and we got pretty worried about him. His T4 numbers, which is thyroxine, the thyroid hormone with 4 iodine atoms in its molecule, were very high. We had 2 options for treatment: a daily pill for the rest of his life, or a one-time injection of a radioactive isotope of iodine called iodine-131. If you've ever tried to give a cat a pill, you'll understand why we chose the radiation treatment. And what that does is it goes in there and destroys a lot of the hormone-producing cells of the thyroid. So we did that about 10 or 11 days or so ago, and for 2 weeks he has to be quarantined because he's radioactive. So we can't let him near our other pets or our kids, and we can't really spend too much time with him cuddling and sleeping and things like that because it puts us at risk. And all of his waste, his pee and his poop, have to be disposed of separately from everything else and stored in a bin outside for 2 months before it can be put in the regular trash. This is so that isotope has a chance to decay enough to not generate positive radioactivity in the trash dump. So it's been quite an interesting ride here. The poor cat is very lonely and is used to spending a lot of time with us. In fact, we found him on the street in the summer of 2004 when he was about 2 weeks old. He was abandoned by his mother and struggling to walk across the street. So I pulled my car over, I stopped traffic, and I picked him up. My wife and I took him to the Humane Society, and they said he was too young for them to take care of. They don't have the personnel, and they would euthanize him. So we decided to keep him. We called our sister-in-law, who's a vet, and she told us all the things you need to do for a kitten that young. Bottle feeding, stimulating them to go to the bathroom, keeping a bottle of hot water in their carrier to keep them warm, all those things. I had to bring the cat to work with me because he needed to be bottle fed every 2 hours, like a human baby. We ended up naming him Getty, after the bass player and singer for the Canadian rock band Rush, which is my favorite band. So now Getty is 15.5 years old, and I think this radiation treatment will give him a few more years with us. Okay, thanks for listening to my story about my beloved cat. Let's get into today's A&P content. In today's episode, we're going to step inside a human cell and explore the cytoplasm the way a biology major would. Not as vague cell stuff, but as a dense, organized world of fluid chemistry, structural scaffolding, molecular machines, and membrane-bound compartments that divide labor with incredible efficiency. If you imagine the cell as a living city, the cytoplasm is everything inside the city walls, but outside the central archives. Crowded streets of reacting molecules, factories assembling proteins and lipids, shipping centers labeling and routing cargo, recycling plants breaking down worn-out parts, detox facilities neutralizing dangerous chemicals, and power stations that run the whole operation. Along the way, I'll make it clear which components are membranous and which are not, and I'll connect each one to an example of clinical significance so the material feels relevant rather than abstract. To begin, it helps to define what we mean by cytoplasm. In most anatomy and physiology contexts, cytoplasm refers to the region between the plasma membrane and the nucleus. It includes the cytosol, which is an intracellular fluid, and the organelles and structural elements suspended within it. Some of these organelles are surrounded by membranes, which create chemically distinct compartments. Others are non-membranous protein assemblies that function more like tools or scaffolds than rooms. That difference is not just anatomical trivia, it is the reason the cell can run incompatible processes side by side without chaos. Enzymes that require an acidic environment, for example, can be isolated inside a membrane-bound compartment, while nearby reactions can continue at a neutral pH. Let's start with the most basic component, the cytosol. The cytosol is often described as a watery fluid, but it behaves more like a crowded gel packed with ions, metabolites, enzymes, RNA, and proteins. This molecular crowding is physiologically important because it affects the diffusion and reaction rates. Many essential pathways occur here, including glycolysis, which breaks glucose into pyruvate, and a large number of intracellular signaling, where cascades of phosphorylation events transmit information from the cell surface to targets deeper inside. The cytosol is also the environment in which many proteins are synthesized and begin to fold. Clinically, the cytosol is where cellular balance can tip into crisis. A dramatic example is calcium overload during ischemia. Ischemia is when blood flow is diminished. When ATP runs low, ion pumps fail, cytosolic calcium rises, and calcium-activated enzymes begin damaging membranes and proteins, contributing to irreversible cell injury. Another common clinical thread is metabolic acidosis, which can be driven by increased cytosolic production of lactate when oxygen delivery is inadequate and cells rely more heavily on anaerobic metabolism. Let me explain that for a second. Anaerobic fermentation is how we produce ATP when oxygen or mitochondria are not present. It is the counterpart of aerobic respiration. One of the byproducts of anaerobic fermentation is lactate. The problem is that lactate is produced when lactic acid ionizes, shedding a hydrogen ion. And when hydrogen ion concentration increases, pH drops, meaning it becomes more acidic. Since this acidity is a result of a metabolic issue, it is called metabolic acidosis. Suspended in this cytosol are ribosomes, the cell's protein-building machines, and ribosomes are non-membranous. Structurally, they are complexes of ribosomal RNA and proteins that translate the genetic message carried by messenger RNA into an amino acid sequence. We're going to talk more about this in detail in subsequent episodes. Ribosomes come in two functional settings: free in the cytosol or bound to the rough endoplasmic reticulum. This location matters because it correlates with where the newly made protein is headed. Proteins made on free ribosomes generally remain in the cytosol or are imported into the nucleus or mitochondria. Proteins made on ribosomes attached to the rough endoplasmic reticulum are typically destined for secretion, insertion into membranes, or delivery to lysosomes. Clinically, ribosomes matter in ways that might surprise students at first. Many antibiotics target bacterial ribosomes, and because mitochondria share evolutionary ancestry with bacteria, some drugs can unintentionally interfere with mitochondrial protein synthesis and ATP production. This becomes most apparent in tissues with high ATP demand. Inherited disruptions in ribosome biogenesis and function, often called ribosomopathies, can also cause problems in rapidly dividing tissues like bone marrow because those cells have little tolerance for impaired protein production. When ribosomes attach to the endoplasmic reticulum, we enter the world of membranous organelles. The rough ER is a network of flattened sacs and membranous sheets called cisternae, and its outer surface looks rough under the microscope because it's studded with ribosomes. The rough ER is central to the synthesis of proteins that must enter the secretory pathway, which means they're going to be secreted out of the cell. As these proteins are translated, they are threaded into the lumen of the endoplasmic reticulum, where they fold into their functional shapes, form disulfide bonds, and undergo early modifications. Just as importantly, the rough ER acts as a quality control checkpoint. Misfolded proteins can be retained and refolded, or they can be targeted for destruction. Clinically, that quality control role becomes a major storyline in disease. When misfolded proteins accumulate, cells experience endoplasmic reticular stress and activate the unfolded protein response. If that stress is chronic, it can contribute to pathology including dysfunction and death of insulin-producing beta cells in type 2 diabetes and cellular stress responses implicated in several degenerative disorders. Another clinically relevant pattern is that certain inherited mutations create proteins that could work in principle, but because they fold incorrectly, they never leave the ER to reach their proper destination at the membrane, effectively creating disease by trafficking failure. The smooth endoplasmic reticulum, or smooth ER, is also membranous but lacks ribosomes and is more tubular in shape. Its functions depend heavily on the cell type. In many cells, smooth ER is a major site of lipid synthesis, producing phospholipids for membranes and contributing to cholesterol metabolism. In steroid-producing tissues, such as the adrenal cortex and gonads, smooth ER becomes abundant because steroid hormones are synthesized from lipid precursors. In liver cells, or hepatocytes, smooth ER contains enzyme systems that modify drugs and toxins, often making them more water-soluble so that they can be eliminated. In muscle cells, a specialized form of smooth ER, called the sarcoplasmic reticulum, stores calcium ions and releases them to trigger muscle contraction. Clinically, smooth ER is tied to drug metabolism and to calcium-dependent physiology. Chronic exposure to certain drugs can induce expansion of smooth ER in hepatocytes, changing how rapidly medications are broken down and sometimes altering effective dosing of those medications. While in muscle and other excitable tissues like neurons, abnormalities in calcium handling proteins associated with the sarcoplasmic reticulum can produce serious disorders of muscle contraction and metabolic regulation, reminding us that one organelle's dysfunction can result in a whole organ's dysfunction. Proteins and lipids synthesized in the ER are often sent to the Golgi complex, another membranous organelle composed of stacked, flattened sacs. The Golgi is frequently described as the cell's post office, and that analogy fits because its job is to modify, sort, and package molecules for their destinations. Material enters the Golgi at the cis face, which is oriented toward the ER, and exits from the trans face, where transport vesicles bud off for delivery to the plasma membrane. That plasma membrane will then secrete the product outside the cell or route it to lysosomes. In the Golgi, carbohydrates, also known as glycans, may be added to a protein. This process is called glycosylation. Similarly, a sulfate group may be added, a process called sulfation. And the Golgi complex performs other modifications that affect stability, activity, and targeting. Clinically, when Golgi processing goes wrong, particularly glycosylation, cells can mishandle an enormous range of proteins. Congenital disorders of glycosylation can produce a multi-system disease because glycoproteins are central to receptors, signaling, extracellular matrix composition, and immune function. In other words, even if the protein background is correct, the wrong carbohydrate pattern can cause the body to treat a vital molecule as defective or misroute it entirely. One major destination for Golgi processed enzymes and cargo is the lysosomal system. Lysosomes are membranous organelles containing digestive enzymes that function best at an acidic pH. Their role is controlled breakdown and recycling. Lysosomes fuse with vesicles carrying material taken in from outside the cell, and they also participate in autophagy, a process in which the cell encloses worn-out organelles and cytoplasmic components and delivers them for degradation. This is not mere cellular housekeeping, it is a survival strategy that reclaims building blocks and prevents accumulation of damaged components. Clinically, lysosomes are famous because when a single lysosomal enzyme is missing or defective, undigested substrates accumulate and lysosomes swell with stored material. These lysosomal storage diseases often damage neurons and other long-living cells and can cause progressive multi-organ dysfunction. The clinical takeaway here is that a tiny defect in a single enzyme can cripple a whole organ system because the cell's recycling machinery becomes congested and toxic. Now, not all degradation happens in lysosomes. In parallel, the cell uses proteasomes, which are non-membranous protein complexes that act like precision shredders. Proteasomes degrade proteins that have been tagged with ubiquitin, a small protein that serves as a molecular label for disposal. This system is critical for removing misfolded or damaged proteins, but it also serves as a powerful regulatory tool. Many key cellular regulators are short-lived by design, and the proteasome controls their levels tightly. This shapes processes like cell cycle progression and signaling pathway activation. Clinically, proteasomes matter because cancer cells often have high protein turnover and intense cellular stress, making them particularly dependent on proteasome function. Therapeutic inhibition of proteasomes can push malignant cells over the edge into apoptosis, which is programmed cell death. Conversely, failure to clear abnormal proteins effectively is a recurring theme in degenerative disease, where toxic proteins can accumulate when degradation systems are overwhelmed or impaired. Another membranous organelle dedicated to detoxification and lipid processing is the peroxisome. Peroxisomes carry out oxidation reactions, including the breakdown of very long-chain fatty acids and the detoxification of certain harmful compounds. A key feature of peroxisomal metabolism is the production of hydrogen peroxide, a reactive molecule that can be dangerous to cells. Peroxisomes therefore house enzymes such as catalase that neutralize hydrogen peroxide and limit collateral damage. Clinically, peroxisomal disorders can be severe because certain lipids accumulate and disrupt membrane composition and myelin integrity in neurons. This creates profound effects on the nervous system and development. These diseases underscore that detoxification and specialized lipid processing are not optional add-ons. They are central to normal organ function and growth. If we zoom out from these single membrane organelles, we come to mitochondria and the nucleus, both surrounded by double membranes. Mitochondria are often called the powerhouse of the cell, and while that phrase can become cliché, it does reflect a truth. Mitochondria are the dominant source of ATP in most aerobic tissues through oxidative phosphorylation. Each mitochondrion has an outer membrane and an inner membrane that folds inside the organelle into these folded structures called cristae. They expand the surface area for the electron transport chain and ATP synthase. Inside the inner membrane is the matrix, which contains enzymes for metabolic pathways and, importantly, mitochondrial genetic material. Mitochondria also influence apoptosis and cellular signaling, and they generate reactive oxygen species as byproducts of electron transport. Reactive oxygen species, or ROSs, are molecules that can contribute to oxidative damage but also serve roles in regulated signaling. Mitochondrial DNA is one of the most fascinating features of human cell biology because it points backward in evolutionary time. Mitochondria contain their own small genome, encoding some mitochondrial proteins as well as ribosomal RNAs and transfer RNAs, which are required for mitochondrial protein synthesis. We will cover these RNAs in subsequent episodes. This fact supports the endosymbiotic theory, in which mitochondria are thought to be descendant from bacteria-like organisms that entered into a symbiotic relationship with early eukaryotic cells. Remember, eukaryotic cells are those that have a nucleus, membrane-bound organelles, and linear DNA. These include plant, fungus, and animal cells, like humans. This is opposed to prokaryotes, which are single-celled organisms that have circular DNA, no nucleus, and no membrane-bound organelles, like bacteria and archaea. Over evolutionary time, many genes were transferred to the nucleus, but mitochondria retained a subset, along with other bacteria-like features. Clinically, mitochondrial DNA matters because mutations in mitochondrial DNA can compromise ATP production, and tissues with high energy demands like the brain, skeletal muscle, and heart are often hit the hardest. These disorders can present with combinations of neuromuscular weakness, exercise intolerance, cardiomyopathy, seizures, and multi-system involvement. Another clinically important twist is inheritance. Mitochondrial DNA is typically maternally inherited, shaping patterns of transmission and genetic counseling in families with mitochondrial disease. Depending on where you're studying A&P, you may be learning about how the mitochondria synthesize ATP while you discuss the cytoplasm and organelles. Sometimes instructors wait until the metabolism chapter, much later in the course. But I want to cover all the bases, so I think we should discuss it here. We have time. It starts with carbohydrate metabolism. Carbohydrates are broken down into monosaccharides and absorbed by our digestive system. Ultimately, the ones that aren't already glucose are converted to glucose. In the cytosol, glycolysis converts the glucose into 2 molecules of ATP, 2 molecules of pyruvate, and some other factors I'll mention in a few minutes. What happens to pyruvate depends on whether or not oxygen or mitochondria are present. If they're not, we go to anaerobic fermentation in the cytosol. If we do have mitochondria and plentiful oxygen, pyruvate enters the mitochondria and undergoes aerobic respiration. Let's talk about all of these steps. Most dietary carbohydrate is burned as fuel within a few hours of absorption. Although 3 monosaccharides are absorbed from digested food, glucose, galactose, and fructose, the last 2 are quickly converted to glucose, and all oxidative carbohydrate metabolism is essentially a matter of glucose catabolism. That's the breakdown of glucose. The overall reaction for this process is C6H12O6, that's glucose, plus 6 oxygen molecules, O2, yields 6 carbon dioxide molecules, CO2, and 6 water molecules, H2O. The purpose of this reaction isn't simply to generate carbon dioxide and water, but to transfer energy from glucose to ATP, adenosine triphosphate, the molecular unit of energy transfer in cells. To understand this energy transfer, recall that oxidation is a chemical reaction in which a molecule, atom, or ion loses electrons. This loss is also described as an increase in the oxidation state of the molecule, atom, or ion. Although often associated with the gain of oxygen, oxidation can occur independently of oxygen. Its counterpart is reduction, the gain of electrons. In the pathway of glucose oxidation, several points exist where other nutrients, especially fats and amino acids, can also be oxidized to generate fuel. This makes carbohydrate catabolism a central reference point from which the catabolism of all fuels and the generation of ATP can be understood. If the overall glucose oxidation reaction were carried out in a single step, it would release a sudden burst of heat, similar to burning paper, for example, which follows the same chemical equation. Such a release would be useless to cellular metabolism and would be harmful, potentially lethal, to our cells. However, the body executes this reaction through a series of steps each catalyzed by a different enzyme. This approach allows energy to be released in manageable amounts, with as much of it as possible transferred to ATP. The remaining energy is dissipated as heat. There are three major pathways of glucose catabolism. One is glycolysis. Glycolysis splits one glucose molecule into two molecules of pyruvate. Two is anaerobic fermentation, which reduces pyruvate to lactate in the absence of oxygen. Anaerobic means without oxygen. And three, aerobic respiration, which requires oxygen and oxidizes pyruvate to carbon dioxide and water. Enzymes catalyze the removal of electrons in the form of hydrogen atoms from intermediate compounds, but do not retain them. Instead, these hydrogen atoms are transferred to compounds called coenzymes, which later donate them to other compounds in subsequent reactions. Thus, the enzymes of glucose catabolism depend on coenzymes for function. Two coenzymes are especially important. NAD+, which stands for nicotinamide adenine dinucleotide, and FAD, which stands for flavin adenine dinucleotide. NAD+ is derived from niacin, which is vitamin B3, and FAD from riboflavin, which is vitamin B2. You may hear people informally say NAD+ and FAD, rather than individually saying the letters of these acronyms. I'm going to say the letters out to avoid confusion. But listen closely to your instructors, as they may say NAD+ and FAD, and I don't want you to get confused. Okay, so hydrogen atoms are removed in pairs, 2 protons and 2 electrons, and then transferred to a coenzyme. This produces a reduced coenzyme with a higher free energy content. The reactions are FAD plus 2H yields FADH2, and NAD plus 2H yields NADH plus a hydrogen ion. FAD binds both protons and electrons to become FADH2. NAD plus, in contrast, binds the 2 electrons but only 1 proton to become NADH. The other proton remains a free hydrogen ion, or in solution, H3O+. Okay, let's look at the first catabolic process for glucose, glycolysis. Upon entering the cell, glucose is subjected to a series of conversions known as glycolysis. This pathway involves the following 5 steps: phosphorylation, priming, cleavage, oxidation, and dephosphorylation. Let's talk about those individually. The first phase, phosphorylation, is when the enzyme hexokinase transfers a phosphate group from ATP to glucose, producing glucose 6-phosphate, or G6P. This traps glucose inside the cell and maintains a concentration gradient that favors continued glucose diffusion into the cell. Phosphorylated compounds cannot pass through the plasma membrane, so that prevents glucose from leaving. In most cells, this step is irreversible due to the absence of the enzyme that reverts G6P back to glucose. Exceptions include the absorptive cells of the small intestine, proximal convoluted tubule cells of the kidney, and liver cells, or hepatocytes. Glucose 6-phosphate is a versatile intermediate that can be converted to fats, amino acids, glycogen, or be further oxidized for energy. The second step is priming. Glucose 6-phosphate is isomerized to fructose 6-phosphate, which is again phosphorylated to form fructose 1,6-diphosphate. This primes the molecule, similar to lighting a fire with a match. 2 ATP molecules are consumed, but aerobic respiration will ultimately yield far more ATP. So even though we're going to lose 2 molecules of ATP during the priming phase, we're going to make so much more ATP during aerobic respiration that will make up for it with tons to spare. Okay, the third step is cleavage. That fructose 1,6-diphosphate splits into two 3-carbon molecules. After a slight rearrangement, both become PGAL, phosphoglyceraldehyde or glyceraldehyde 3-phosphate, either one of those. The fourth step is oxidation. Each PGAL is oxidized by removing a pair of hydrogen atoms. 1 proton and 2 electrons are accepted by NAD+, forming NADH plus our H+, our hydrogen ion. A phosphate group is added to each 3-carbon intermediate from the cell's phosphate pool, not from ATP. And then finally, dephosphorylation. In the final step, phosphate groups are transferred from the intermediates to ADP, adenosine diphosphate, forming ATP, adenosine triphosphate, and converting the intermediates into pyruvate. The net products of glycolysis from one glucose molecule are 2 pyruvate plus 2 NADH plus 2 hydrogen ions plus 2 ATP. While 4 ATP are produced in the dephosphorylation step, 2 were consumed in the initial phosphorylation and priming steps. So that means we have a net gain of 2 ATP. Energy still remains in the form of NADH and pyruvate, though. That's when we get into anaerobic fermentation. What happens to pyruvate is going to depend on whether or not oxygen is available. In oxygen-deprived conditions, such as intense muscular activity, cells resort to anaerobic fermentation. This allows for limited ATP production when oxygen is scarce or when mitochondria are absent, like we have in red blood cells or erythrocytes. However, glycolysis alone would quickly halt if pyruvate were not metabolized. This is because NAD+ is consumed during glycolysis and must be regenerated. Anaerobic fermentation solves this by reducing pyruvate to lactate using electrons from NADH, thereby regenerating NADH+. Lactate exits the cell and travels via the bloodstream to the liver. When oxygen becomes available again, the liver oxidizes lactate back to pyruvate, which may then enter aerobic pathways. This oxygen-dependent recovery is part of the excess post-exercise oxygen consumption, or EPOC. The liver may also convert lactate to G6P, glucose 6-phosphate, and either store it as glycogen or remove the phosphate to release free glucose into the blood. Despite its usefulness, anaerobic fermentation has drawbacks. It is inefficient since most energy remains in lactate and contributes to no useful work. And lactate is toxic in excess. Skeletal muscle tolerates this better than cardiac muscle, while the brain employs anaerobic fermentation minimally. During birth, however, most fetal organs switch to anaerobic metabolism to preserve oxygen for the brain. Okay, then we get to aerobic respiration, and that's when it gets really, really intricate. When oxygen is plentiful, pyruvate enters the mitochondria for aerobic respiration, which includes 2 phases: the matrix reactions and the membrane reactions. So let's talk a little bit about mitochondria, because you might remember from your classes or way back in the very early episodes of this podcast that mitochondria are the powerhouse of the cell. They're the cells that synthesize ATP for us. They're the ones that are giving us the most ATP. That's because they're responsible for aerobic respiration. Remember that the mitochondrial membrane is a double-layer membrane. There's an outer membrane and an inner membrane. The inner layer folds into the inside of the mitochondria. Those infoldings of the inner mitochondrial membrane are called the cristae, which is the plural of crista. The space between the cristae is called the matrix. So we have some reactions that take place in the matrix. And we have some reactions that actually take place in the inner mitochondrial membrane, the cristae. So we have matrix reactions and we have membrane reactions. And the main process that we're going to see happen in these mitochondria is the citric acid cycle, which you may have heard as the Krebs cycle. It constitutes most of the matrix reactions, but several steps have to take place before the citric acid cycle. So let's talk about those. First, Pyruvate undergoes decarboxylation, losing one carbon as CO2 and forming a two-carbon compound. NAD+ oxidizes this compound, forming an acetyl group. The acetyl group binds to coenzyme A, which comes from pantothenic acid, to form acetyl-CoA, which is ready to enter the citric acid cycle. Acetyl-CoA donates 2 carbons to oxaloacetic acid, which has 4 carbons, forming citric acid, which has 6 carbons. Citric acid is rearranged and retains 6 carbon atoms. Hydrogen atoms are removed and accepted by NAD+. Another carbon dioxide, CO2, is removed, leaving a 5-carbon intermediate. Then those last 2 steps repeat. Hydrogen atoms are again removed and accepted by NAD+, and another CO2 is removed, leaving a 5-carbon intermediate. That generates another carbon dioxide and yielding a 4-carbon intermediate. At this point, all 3 carbons from pyruvate have been lost as carbon dioxide, CO2. Energy is transferred to GDP to form GTP, which donates a phosphate to ADP, adenosine diphosphate, to make adenosine triphosphate. FAD accepts 2 hydrogen atoms. Then water is added to the system, and 2 hydrogen atoms are transferred to NAD+, forming oxaloacetic acid, completing the citric acid cycle. So for each glucose molecule, these matrix reactions occur twice because there were 2 pyruvates. Okay, the matrix reactions can be summarized in 4 different equations. The first one is 2 pyruvate plus 6 waters yields 6 carbon dioxide. The second is 2 ADP plus 2 phosphates yields 2 ATP. The next is 8 NAD plus 8 hydrogen molecules, H2, yields 8 NADH plus 8 hydrogen ions, or protons. And then the last one is 2 FAD plus 2 hydrogen atoms plus 2 hydrogen molecules, H2, yields 2 FADH2. At this stage, the original glucose's carbon atoms have all been released as CO2. While some energy is lost as heat, a significant portion is captured in 2 ATP, 8 NADH, and 2 FADH2 molecules. The citric acid cycle is amphibolic, which means it has both catabolic and anabolic functions. It's not only breaking down glucose, it's producing intermediates for synthesizing fats, non-essential amino acids, and heme. Okay, that was the matrix reactions. Now we have the membrane reactions, which are electron transport and ATP synthesis. The membrane reactions serve 2 purposes. One is to further oxidize NADH and FADH2, transferring their energy to ATP. 2 is regenerate NAD+ and FAD for reuse. These steps occur through the mitochondrial electron transport chain composed of flavin mononucleotide, which receives electrons from NADH, iron sulfur centers, coenzyme Q, which accepts electrons from FADH2, copper ions, and cytochromes. Hydrogen atoms are split. Protons are pumped into the intermembrane space and electrons pass through the chain. Each carrier becomes reduced when it gains electrons and oxidized as it passes them along. Oxygen serves as the final electron acceptor. 1/2 O2 plus 2 electrons plus 2 protons yields water, H2O. This reaction forms metabolic water and underscores the body's need for oxygen. If oxygen is unavailable, the chain halts, stopping ATP production and leading to cell death. Then we have the chemiosmotic mechanism. It's called chemiosmosis because it's similar to osmosis but involves chemical substances instead of water. Chemiosmosis is the process where ions, typically protons like hydrogen ions, move across a membrane down their electrochemical gradient, releasing energy that is used to synthesize ATP. The electrons moving down the transport chain liberate energy, some of which is lost as heat, but some is used to fuel three respiratory enzyme complexes that involve the components of the electron transport chain. Those respiratory enzyme complexes are 1. Flavin mononucleotide, or FMN, and multiple iron sulfur centers, 2. Cytochromes B and C1, and an iron sulfur center, and 3. 2 copper centers and cytochromes A and A3. These complexes are proton pumps, removing hydrogen ions from the matrix and pumping them into the space between the inner and outer mitochondrial membranes. Coenzyme Q transports electrons from complex 1 to complex 2, while cytochrome C transports them from complex 2 to complex 3. You may have heard of coenzyme Q10 as a dietary supplement. One of the reasons people take CoQ10 is because cholesterol-reducing drugs called statins are known to reduce CoQ synthesis, resulting in muscle pain and weakness. While the level of hydrogen ions in the matrix declines, creating an electrochemical gradient across the inner mitochondrial membrane and the matrix, hydrogen ions can only cross the membrane via ATP synthase pumps. As they follow through these pumps down their electrochemical gradient, a current is generated, whose energy is harnessed by the enzyme ATP synthase and used for ATP synthesis. This process is called chemiosmosis. Protons accumulate between the inner and outer mitochondrial membranes, establishing an electrochemical gradient. This gradient is harnessed by an enzyme called ATPase synthase, a channel protein that allows hydrogen ions to flow back into the matrix, generating ATP. This mechanism is called chemiosmosis. Okay, so what is our ATP yield from glucose oxidation? Each NADH yields approximately 2.5 ATP, and each FADH2 yields about 1.5 ATP. So if we look at the math, we have a total of 10 NADH that we're going to get ATP from. 2.5 each, that's 25 ATPs. We have 2 FADH2, and we're going to get 1.5 ATP out of each of those. That's 3. So now we're at 28. Glycolysis gives us 2. Now we're at 30. And the matrix reactions give us 2. That gives us a total of 32 ATP per glucose. This is a theoretical maximum. There's variability based on the shuttle mechanism used by different tissues. And those shuttle mechanisms are malate versus glycerol phosphate, and the energy required to transport ATP out of the mitochondria. This all varies. For example, in the cells of the kidneys, liver, and cardiac muscle, NADH transfers its electrons to malate. However, in the brain and skeletal muscle cells, NADH transfers its electrons to glycerol phosphate. Less ATP is generated by the glycerol phosphate shuttle, and there's still cells for which the ATP yield is completely unknown. So different tissues are going to have different yields for ATP, but this is a good general idea of what we're getting out of a glucose molecule if oxygen is available.
The secret is out. With millions of younger-looking customers across the country, Plexiderm is one of the most highly sought-after products on the market. It visibly shrinks under eye bags and smooths out crow's feet in minutes, and the effects last up to 10 hours. Influencers love it, magazines feature it, and people just can't get enough of it. As a matter of fact, Elle magazine gave it an Editor's Choice Award as Best Instant Wrinkle Filler for 2025. In as little as 10 minutes, Plexiderm reduces the appearance of fine lines, wrinkles, and those dreaded under-eye bags. So yes, you can look years younger in minutes, and the effects last for hours. No surgery, no gimmicks, just results. The best news, you can try Plexiderm for only $14.95. Plus, get free shipping. Don't wait. Order Plexiderm today. Visit plexiderm.com/special. That's plexiderm.com/special. Paid for by Sheer Science.
The nucleus, while technically distinct from the cytoplasm, is inseparable from cytoplasmic function because it directs much of what the cytoplasm does, and the cytoplasm supplies many of the nucleus's building blocks and machinery. The nucleus is enclosed by a double membrane nuclear envelope perforated by nuclear pores that regulate traffic in and out of the nucleus. Within the nucleus, DNA is packaged with proteins into chromatin, and the nucleolus is the site where ribosomal units begin assembly. The nucleus stores and protects genetic information, carries out transcription to produce RNA, and coordinates gene expression programs that determine a cell's identity and behavior. Clinically, defects in nuclear structure and regulation can be devastating. Alterations in nuclear envelope proteins can weaken cells that experience mechanical stress, such as muscle cells, and disruptions in nuclear control of the cell cycle and DNA repair are central to cancer biology. We're going to talk much more about the nucleus and the functions that take place inside of it and the functions it dictates outside of it in the cytoplasm in the next couple of episodes. Holding all of this together is the cytoskeleton, a non-membranous system of protein filaments that gives the cell structure, allows movement, and organizes transport. The cytoskeleton is dynamic. It is constantly assembling and disassembling as the cell changes shape, migrates, divides, and moves cargo. Microfilaments made of actin support the cell cortex beneath the membrane, contribute to cell motility, and, when paired with myosin, generate contractile forces. Intermediate filaments provide tensile strength, helping cells resist stretching and pulling. They're especially important in tissues that endure mechanical stress. Microtubules, which are hollow tubes made of tubulin, function as tracks for long-distance intracellular transport and are essential for the mitotic spindle during cell division. Clinically, cytoskeletal defects show up in a wide range of ways. Mutations in keratin intermediate filaments can produce fragile skin, which is prone to blistering because cells cannot withstand mechanical stress. Drugs that target microtubules can halt cell division, which is useful in cancer therapy, but they can also injure rapidly dividing tissues and contribute to side effects. Disorders of the microtubules of cilia can impair mucociliary clearance in the respiratory tract, predisposing us to chronic infections, an example of how microscopic structure can shape organ-level disease. Microtubule organization leads naturally to the centrosome, the cell's primary microtubule organizing center, and the centrioles that are housed within it. The centrosome is non-membranous and contains a pair of centrioles arranged roughly at right angles. They're embedded in a protein-rich matrix. Its main job is to nucleate and anchor microtubules, organizing the cell's internal structure and becoming especially critical during cell division when it helps form the mitotic spindle. Centrioles also relate to basal bodies with anchor cilia. Clinically, centrosome dysfunction can contribute to chromosomal missegregation and genomic instability, patterns frequently associated with cancer. When spindle organization fails, daughter cells can acquire abnormal chromosome numbers, and that instability can drive malignant progression. Okay, finally, we should talk about inclusions. These are intracellular accumulations that are not usually considered organelles. Inclusions often represent stored nutrients or pigments, and many are non-membranous, although some, like lipid droplets, have specialized boundary layers. Glycogen granules store glucose in polymer form for rapid mobilization. Lipid droplets store energy-rich triglycerides. Pigments like melanin contribute to coloration and photoprotection, while lipofusin accumulates over time as a wear and tear pigment in long-lived cells. Clinically, inclusions can be clues to metabolic imbalance or disease. Excess lipid storage in hepatocytes can cause fatty liver disease, which can progress from steatosis to inflammation and fibrosis of the liver. Abnormal glycogen accumulation is seen in glycogen storage diseases, where defects in enzymes for glycogen synthesis or breakdown lead to organ dysfunction, particularly in the liver and the muscle cells. When you step back and look at the cytoplasm as a whole, you can see why the distinction between membranous and non-membranous structures is so powerful. Membranes create compartments that allow the cell to maintain different microenvironments: acidic interiors for digestion, specialized lumens for protein folding, segregated spaces for oxidation reactions, and electrochemical gradients for ATP production. Non-membranous structures, by contrast, are the cell's machines and framework: ribosomes that build, proteasomes that dismantle, cytoskeleton that shapes and transports, and organizing centers that coordinate division and movement. So as we close, remember that the cytoplasm is not empty space. It is a densely coordinated ecosystem where structure serves function at every scale. The cytosol hosts metabolism and signaling. Ribosomes build proteins, the rough ER folds and checks those proteins, the smooth ER synthesizes lipids and detoxifies chemicals, the Golgi modifies and routes cargo. Lysosomes and proteasomes keep the cell clean by breaking down what is damaged or no longer needed. Peroxisomes handle specialized oxidation and detox reactions. Mitochondria power the system and carry a genetic trace of ancient symbiosis, while the nucleus safeguards the genome and directs cellular identity. The cytoskeleton and centrosome provides organization, motion, and division, and even inclusions, simple as they seem, reflect the cell's nutritional status and its history. Okay, that should do it for the cytoplasm. Once again, thank you for listening to my podcast. I'll be sure to keep you posted on my cat's health as we move on. If you have a question, please email me at minus55media@gmail.com. I hope this podcast is helping you get your B or better in A&P. Good luck, be well, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media and is hosted by the Believe Network. Please take the time to rate my show and leave a review. You can also check out the content I post on Instagram and YouTube as @StudentHelp4AP. That's the number 4 AP. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where your instructor can find Anatomy and Physiology Digital Suite, my low-cost tutor video-based digital learning solution for anatomy and physiology currently being used at several colleges and universities. If your instructor isn't using Digital Suite, let them know about it. The link is in the show notes.
If you like the show, please take a moment to rate, review, and subscribe. It really does help the show to grow. Thank you for listening.